Perwakilan yang akan mempresentasikan adalah perwakilan dari kelompok riset psikologi. Nanti akan ada kolega dari eh, Kaketris yang akan menyampaikan. Namun sebelum kita memulai, sebelum acara saya serahkan kepada moderator, sebelumnya akan ada sambutan terlebih dahulu dari Kepala Pusat Riset Biosistematik dan Evolusi kepada Pak Arif. Dipersilakan, Pak.
Baik, terima kasih Mbak Gloria, Bapak Ibu semua. Assalamualaikum warahmatullahi wabarakatuh. Selamat
Assalamualaikum warahmatullahi
sore Bapak
wabarakatuh.
Ibu, selamat datang kembali di SOS Pusat Riset Biosistematika dan Evolusi yang ke-72, yang kali ini bagiannya alga begitu ya. Dengan topik sepertinya sangat menarik begitu, diatom, tentang diatom dari basic research sampai dengan bioprospecting. Jadi hari ini kita kedatangan tamu. Good afternoon from Indonesia and very good morning to you, Dr. Dabek ya, in
Hi,
Poland.
hello, thank you
Yes,
for the invitation.
yes.
Good
Welcome
afternoon.
to our scientific talk in Research Center for Biosystematic and Evolution. So maybe first of all, thank you very much for accepting our invitation to share your research experience with us today. We truly look forward to discussing with you, especially for our group. And we hope this will be the beginning of our fruitful collaboration in the future, hopefully, yeah. In the future, we would like very welcome to you if you interest to visit Indonesia to discuss with us face to face and conducting the discussion and potential joint research if possible. So please let us know if you have time to visit Indonesia. It is far away. Then
Oh,
maybe yes,
anytime.
yes, great. Then Pak Vidi and then team and also myself will preparing for it. So we invite you if you have time, please come to Indonesia. So no, uh, talking about the diatom, maybe I have no experience about that, but maybe this is very interesting because this organism is incredible important, yeah, for the world. Diatom, as we know, that responsible for producing like approximately 20 up to 50%, yeah, of the oxygen productivity, yeah, from photosynthesis. I check from the Google.
Yeah,
So
correct.
they form the base of aquatic food web and play a major role of the global carbon and then serve as the valuable indicator for the water quality and climate change. Diatom are the relative young group, as I know. In the term of evolution history, they are considered for the most successful unicellular phytoplankton in the ocean, with the estimation diversity, as I know, approximately 100,000 species or more, then
Oh,
highly
these are
diverse,
a lot
more
of
than,
different estimates. Yeah.
yeah, 100,000, yeah, and only approximately 12,000 species or less than 12% have been described. But the problem in diatom is probably the morphology similarity, and then vast number of undescribed species, limited molecular database, and then complex structure required expert analysis, like you probably. When in the come to bioprospecting, the major bottleneck, as I know, is, for example, the low biomass productivity in dense culture, which limit to commercialize for the application like a biofuel and biomaterial. So this complex genome are not easy to be manipulated. The problem in our group in Indonesia, especially, we are deeply concerned about the lack of comprehensive data on diatom diversity from the huge number of known species. How many found in Indonesia, we don't know. How many novel species remain undiscovered in Indonesia? Most of this remaining unknown. So we hope in the future microalga group under Pak Vidi team can help us address this question by the, for example, the dedicated research in the future. So once more, thank you so much for coming, Dr. Debek, and for joining our sharing season this afternoon. We truly appreciate you your time and contribution to our knowledge sharing system today. So terima kasih banyak Bapak Ibu telah bergabung sekali lagi di weekend sore hari ini, baik dari PRBE dan luar PRBE begitu. Semoga dapat menambahkan ke kita hasanah, terutama ilmu tentang diatom begitu ya, karena kita tahu bahwa ini adalah kelompok mikroba yang sangat kecil tetapi memiliki peran yang sangat besar, memiliki kompleksitas yang sangat tinggi juga di taksonomi begitu ya. Jadi kita berharap mudah-mudahan jumlah yang besar itu kita bisa menemukan temuan-temuan baru, terutama yang dari Indonesia. Ini PR besar buat Pak Vidi dan tim begitu ya. Mungkin itu yang bisa saya sampaikan dari kami. Terima kasih banyak. Selamat berdiskusi. Saya akan menyimak dari awal. Terima kasih. Saya kembalikan ke moderator. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Ya, baik. Terima kasih Pak Kapus atas waktu dan sambutannya. Kalau begitu langsung saya serahkan saja acara kepada moderator, kepada Pak Vidi, dipersilakan.
Ya, baik. Terima kasih banyak. Thank you very much, Mbak Gloria. Can you hear my voice clearly?
Yes, clear.
Yes. Okay, thank you, Pak Kapus. Thank you very much indeed, Dr. Arif Nurkanto, for your very warm opening remarks. Yeah, and a very good morning to Dr. Dabek. Good afternoon, and welcome to all attendees joining us from across Indonesia and even beyond. And a very warm welcome to this special sharing session hosted by the Research Center for Biosystematics and Evolution at the National Research and Innovation Agency, BRIN. And it is my distinct pleasure to welcome and introduce our distinguished keynote speaker, Dr. Przemyslaw Dabek. Did I pronounce it right?
Yeah, correct.
Okay, thank you. Actually, we met for the first time in the European Union Horizon 2020, led by Professor Jean-Luc Mouget, our mutual French collaborator, also with our beloved mentor, Prof. Andrzej Witkowski. Yeah. And Dr. Shemek is a world-renowned expert in diatom taxonomy and systematics from the Institute of Marine and Environmental Sciences, University of Stettin, Poland. And his work is really impressive and has fundamentally expanded our understanding in these very microscopic creatures. And if I'm not mistaken, from his CV, he has described countless new species and also genera since 2013, I suppose. Yeah. And authoring many works that are essential reading for anyone in the field of ecology. So his talk today, Diatoms Bridging Basic Research and Bioprospecting, is perfectly poised to inspire our diverse audience from ecologists and microbiologists to students who explore the vast world of biology. So I think it will be interesting because it underscores the incredible journey from fundamental scientific discovery to the real world of application. Yeah. So without further ado, it is my great honor to invite Dr. Shemek to begin his presentation. So Dr. Shemek, the virtual floor is yours. Thank you.
Thank you. Thank you for the invitation and a warm welcome. I didn't expect such a warm welcome. I'm trying to share my screen with you. Tell me if you see the screen.
Yes. Could you make it?
It's coming. Okay.
Okay. Great.
Okay, so good morning and good evening, everyone. My name is Przemysław Dabek. I work with Diatoms for more than 15 years now. Fidi already mentioned some of my CV points. Basically, I start my journey with Diatoms under the supervision of Professor Andrzej Witkowski 15 years ago, and my first work was pure taxonomy and systematics of diatoms. Then with the time, I moved to more applied research, what I'm doing, what I share my time between traditional morphology, traditional biology of diatoms and taxonomy with this applied sciences. So I prepared the talk for today for you, bridging this basic research and bioprospecting, and I tried to prepare the talk as simple as I can so a broad audience can understand the talk. Although if something is not clear enough, then there will be a question session. So please, I'm very happy to answer and to guide you here. Apologize at the beginning because in my building we have some repairments, so you may hear some strange voices over my presentation. So don't be scared. This is unfortunately something which I cannot do anything with this. Okay, let's start my presentation. We are, or me, I'm working in a diatom group of the University of Szczecin. Here we have a simple map. So we are close to the German border, very far away from Warsaw, closer to Berlin. So if you want to visit us, our lab, please fly to Berlin. It's much closer. So what we are doing, basically we cover every topic related to diatoms. So as you see, traditional molecular diatom taxonomy and systematics, genomics, metabarcoding, biogeography, ecophysiology, biomonitoring. Also, this bolded part, which I will be talking about more today, this is the extraction or the production of various metabolites from diatoms and developing of a product, bio-based product on diatoms. These are my folks from our group. This is a current composition of our group. It's changed a lot over the time. We have a colleague from Indonesia, Ella Artsatsulastri. I believe she's present here. We had the collaboration with Indonesia before. I and Fidi mentioned already Andrzej Witkowski. He was a pioneer actually in this collaboration with Poland and Indonesia. But also we had Oktias Lutfi as a PhD student. He graduated just recently, so we have the collaboration with Indonesia, I think, established. So that's me. Here's my email address. You can see also our Instagram page and a Facebook page. So please, you can visit us and follow us on social media as well. So how often do you hear about microalgae? I will start my talk in a broad sense, then we will focus more on diatoms. Why I'm asking you about this? Because we are expecting, or we are coming across with microalgae more and more often recently due to the global climate change. And for example, in the Baltic Sea, so close to my place, this is a very closed marine reservoir with the very limited access to the open ocean. So we see every summer blooms of microalgae, for example, cyanobacterial blooms. You see these green patches all over the screen, which are harmful basically to marine life and to human. They can produce toxic substances, toxins. They can bloom. If they bloom and with a high biomass, they deplete the water with oxygen, with nutrients. This is all related to the high temperatures, to the global climate change, and to, of course, this excessive nutrient release to the ocean, to, for example, Baltic Sea, because of the human activity. So we are struggling more and more recently with this cyanobacterial blooms. You see here, we do monitoring, we do a taxonomy and systematics of these blooms. So with what species they are forming these blooms. And 2 years ago, we had very extensive bloom of cyanobacteria, unexpected one, which took like 20 or 30 days. It covered more or less entire central and southern Baltic Sea. And unfortunately, we discovered that this bloom was caused mainly by these three cyanobacterial species you see here, which are unfortunately toxic. They can produce, for example, Nodularia spomigena, they can produce a nodularine, which is considered as a very strong hepatotoxin, so very harmful to marine life, to marine organisms, as well as a human. So this is a problem, or this is the opportunity for us. I believe it's both, but for example, from this slide, you may see that here, no stock cyanobacteria present also in the Baltic Sea was used from our by our colleagues from the different institute in Poland as a natural bioproduct, a peptide for reducing the replication of coronavirus. So maybe this is actually more opportunity than problem when it comes to microalgae and also diatoms. Microalgae are of course very potential, which I just presented you before, because they have the huge ability to produce high or low value metabolites. These are three groups where we can find some good examples for natural products, promising natural products. I already mentioned cyanobacteria. We have a lot of functional pigments, for example, but also you probably know spirulina, which is very often used in the supplements, food supplements and cosmetics because of some antiviral, anti-cancer properties. The same when it comes to green algae. For example, you probably know or you've heard about chlorella, which has more or less the same properties as spirulina, or Hematococcus pluvialis, which is famous for producing a very strong antioxidant, astaxanthin. I will be talking about this later. Among these microalgae, we also have different candidates producing lipids, so the oil. And this oil can be used in various places in the industry. When it comes to diatoms, diatoms are considered as very good producers for lipids. For example, very rich in the fatty acids like omega-3, 6, or 9. I will be talking about this later on. But also, they are famous for producing fucoxanthin, so the pigment, photosynthetic pigment. But also, I will give you an example of non-photosynthetic pigment called marinin, which is very promising pigment nowadays. Recently, also, we see the huge interest in a fresh gel of diatoms. So this is an exoskeleton made of silica, which can be applied in material engineering and any other branches of industry. Here in the table, you see an example of some biochemical composition of different microalgal groups. You see that various microalgae can produce proteins, carbohydrates, fats in the various amounts. This is very species specific, if I may say that. But I want to give you another example of microalgae, promising microalgae called Botryococcus brownie. Maybe you know these algae already. So what you see here in these two pictures, this is a huge colony of this Botryococcus brownie, the green algae, famous for producing fat, huge fat. All these transparent places here, these granules, sticking to the cells. This is a cell, this is a cell, this is a cell. This is a long chain hydrocarbon and lipids produced by this microalgae, which can be easily and is easily converted to biodiesel. So is it a future? We are talking about the future, or we are talking about present times? I think it's already ongoing. So here you can see the very first commercial plane flight based on biofuel. And on, and year after year, more and more operators, they are shifting, not of course completely, but shifting to more sustainable aviation fuels, for example, from microalgae. There is one company in United States of America producing 100% algae-derived biodiesel. This biodiesel is mainly produced from this Bodriococcus browni, which I just shown you previously. So let's go for a while to a basic research. A basic research is, well, a research to produce knowledge. So what we know about the diatoms. These are golden brown algae. They belong to the kingdom of protists. They have eukaryotic cell, similar to the higher plants or plants in general. So the cell content and organella, they are the same as in the plant cell. They are very small, they are unicellular. You can find diatoms with the size of 1 micrometer, and so not possible to see them with the naked eye, but some of them, they are relatively big, and they can be even up to 2 millimeters, so barely visible with the naked eye. Of course, they are photoautotrophic, which is a very important information to us when it comes to bioprospecting. I already mentioned this hydrated silica wall. You see here this on the screen, a broken part of the frustule. They can live as a solitary cell, or they can form very long and very various in shape colonies. You will find the diatoms everywhere when just a little of sunlight and moisture is available. So, of course, in the aquatic environment, but also in the soil, in the ice, on the trees, you will find also diatoms. This diversity was already mentioned, and I said that there are various estimations how many species are in the world. We can just estimate this, and one of the most recent research saying that we could expect the biodiversity around 200,000 species, which only like less than 10% is described from the various biotopes. What is also important from the bioprospecting point of view, this is the last point. We know that because of the photoautotrophic activity of diatom cell, they are able to produce a lot of different metabolites, which can be used in by human in industry, for example. Okay, so do we still need to know more about diatoms? Of course, I mentioned this huge undiscovered biodiversity, but rest is more or less known about diatoms. So the answer is, of course, yes. Oh yeah? So you may say, okay, prove me that I still need to do taxonomy. I still need to describe new species. So here you are. I prepared the slide, which I will be giving you my personal opinion why the basic research in the biotechnological era still actually matters. So the first point, which I see very interesting, is no application without identification. So you see here that taxonomy and systematics is very important in our understanding of species diversity. I mentioned this 10% discovered species. And this is essential before exploiting this biotechnological potential. We need to have a baseline. We need to know how many species, where they occur. We need to have the accurate classification, which can prevent identification, because this underpins bioprospecting. We need to still learn about this new species morphology and biology, because they will give us an information about the function of some, for example, enzymes or even metabolites. So the structure-function relationship is crucial for the search and use of these biomolecules and biomaterials we can produce based on diatoms. Of course, ecology drives innovation. Why is it like this? Because we may discover diatom, we may describe diatoms, sorry, not discover, but we may describe diatoms, new genera, new species, etc. But we know very little about so-called out ecology. Without out ecology, we cannot go further with bioprospecting. And out ecology is actually a basic research. So this is a knowledge about optimal conditions for this certain species of diatoms, under which conditions this diatom can grow faster, can produce more biomass, or can produce some unique metabolite. Biodiversity, mentioned several times already, is simply bioresource diversity. So without exploring biodiversity of diatoms or any other microalgae, we will not have bioresource diversity. So these, by exploring taxonomic diversity, of course, we expand the portfolio of possible biotechnological applications among microalgae. Physiology builds sustainable use. So when we understand the life cycles, when we understand the metabolic pathways, when we understand physiology of, for example, diatoms, we can prevent producing new biomaterials or bioproducts over exploitations of natural resources, of course. When it comes to genomes, you probably know more than me that genomes, when we dig into the genomes of the organisms, we are revealing very interesting facts. And when we bind this with bioprospecting, we can, of course, we can discover new genes responsible for producing, for example, lipids, etc. So what I want to express here, definitely we need still basic research. We still need this very basic taxonomy study, morphological study, biological study, ecological study, but also we need an integrative approach. By this, I mean we should work together from the people from different fields to have this research well done, let's say. So just a few more explanations before we move further. By bioprospecting, I understand a screening, so the search for a good candidate, like, for example, among diatoms, producing, for example, lipids, producing pigments, to discover and develop these new products, bio-based materials, bio-based products, which can be used further for human. Blue biotechnology is basically an industry based on biotechnological approach to develop and deliver these products based on aquatic organisms, either marine or freshwater, bio-based products, but also processes and services like I will be talking today about, for example, water remediation. So this is a nice example of using diatoms, using blue biotechnology in the way of providing a new services for human. Okay, so let's go back to the lipids again, but now this time let's talk about diatoms. So you see here on the picture various diatom live cells, and inside the cells you see these transparent granules indicated by these arrowheads, and these are lipids. So these are lipid droplets accumulated or excreted from a diatom cell. What you can see here, different diatom species, they produce different, or they are able to accumulate different amount of lipids in their cell. Also here, you will see you see 5 different species from marine diatom genus Halamphora, and you see that even here, these 5 different species, they vary in the way of accumulation of these lipids in the cell. Basically, diatoms are considered as the very important producers among microalgae when it comes to lipids. They can produce highly diverse composition, so many different types of fatty acids. I already told you that this lipids production is very species specific. So you can also see here we have different species and different amount and different composition of lipids. What is good to know from the basic research, and which we can use further in the bioprospecting research, is that under the stress conditions, diatoms can accumulate larger amounts of lipids. By the stress conditions, I mean, for example, nutrient limitation or changes in the light during the cultivation of diatoms. Diatoms are also able to produce highly valued fatty acids. You see 2 types of omega-3 fatty acids, DHA and EPA. Here in this first table, you don't see any diatom. This is just to show you that various microalgae, they are also able to produce this kind of fatty acids. But here in the bottom table, you see some of the diatoms, for example, Nichia leavesi or Phaeodactylum tricornutum or Navicula saprophylla, which are quite efficient in producing this type of fatty acid production. So the question, the fatty acid composition. So the question is, can we now think of a renewable source of these fatty acids, because they have a huge importance on human health? So can we replace diatom or any other microalgal omega-3 fatty acids to fish? So can we replace diatoms? Can we replace fish by diatoms? So currently the pipeline we use in our lab also for the lipids recovery requires killing or destroying the biomass we culture for like several days or weeks. This is basically a culturing. We need to know the ecophysiology of this diatom, so what optimal conditions are for this certain strain. Then we have a step where we dry or we make a powder out of the biomass, and we do the chemical extraction using the organic solvent. And what you see here is a crude oil fraction extracted by the chemical extraction from a live strain. But why not milking? Milking is the process where some diatoms, and in some situations, they can excrete the lipids outside the cell. Like I shown you the example of Botryococcus brownie excreting this pure fat outside the cell. Diatoms, they can also do that under certain circumstances. Of course, we can motivate diatoms to release this oil outside the cell by, for example, electric field stimulation, by ultrasounds, by centrifugations, etc., etc. Milking approach in producing of lipids or any other metabolites is, the advantage of this is that we are not killing diatoms, but we simply taking out the metabolites, the product of our desire. So here is our research. We had 3 strains of Halamphora from different regions. You see strain 1, strain 2, strain 3. What you can see here at the very first look, they are pretty similar when it comes to the shape and also morphology. Of course, they are 3 different species, but also they were able to accumulate various amount of lipids. You see these transparent granules all over inside the cell. So we know that under the standard culture conditions, this strain, this could produce or could accumulate actually 20 to 20% of the lipids of the total cell volume. But when we applied temperature stress, so we lowered the temperature, but first we need to know what is the optimal temperature for growth. But then we lowered the temperature for a while, and under these stress conditions, this strain could accumulate up to 60% of the lipids inside the cell. We did the fatty acids profiling. You see here the table presenting over 20 different fatty acids. They are divided into 3 groups. The most desirable ones are the last group, and you see here that this strain under this environmental stress conditions could produce a lot of these polyunsaturated fatty acids. Over 1/3 of these fatty acids were these PUFAS categories, and inside this over 28% of these polyunsaturated acids was composed by omega-3 fatty acids called ALA. I introduced you these 2, DHA and EPA. Maybe this strain is not very sufficient in producing EPA and DHA omega-3 fatty acids, but at least some are in quite decent amount produced by this species. Together with our Chinese colleagues, we also applied different type of the stress. So you see here, under the normal culture conditions, this Halamphora was able to accumulate, let's say, around 30% of lipids, total lipids, in the dry weight. And among these, around 10% of these triaglycerols, TAGs. But when we applied a nutrients stress, this is a nitrogen stress, it means we depleted the culture medium with a nitrate. So low concentration of nitrogen. This Halamphora strain could accumulate over 40% of total lipids, but out of these, 25% of these triaglycerols. These TAGs fatty acids are considered or more desirable for biofuel application. Frankly saying, diatoms are not very considered as a very promising organisms for biofuel application, because, for example, we have this Botryococcus brownie. But you see here, when we applied the temperature or salinity stress, we could get more omega-3 and omega fatty acids. So we consider diatoms not only as a producer for biodiesel application, but for any other. You see here, I'm presenting you a research of me and my colleagues on this Halamphora strain. When we used this crude oil from Halamphora, we mixed this Halamphora lipids together with different lipids and oils from microalgae, different microalgae, and we formed lipids nanocarriers, lipids nanoparticles around 200 nanometers in size. And by the characterization of these nanolipids, we could say that because of this positive charge, these nanoparticles based on microalgal oil like Halamphora strain, we get the situation when these nanolipids could bound with the negatively charged skin. So this is a possible application for pharmaceuticals and cosmetic industries. We obtained a patent on this, except for the research. We are continuing our research with the different strains and producing different kinds of these lipids and nanoparticles. So we achieved the situation when the lipids from diatoms and different fatty acids compositions from the different microalgae formed a new product, a new bio-based product, nanoparticles, which can be used in pharmaceutical or cosmetics industry. Diatoms are able to produce many different bio products. For example, I had a chance to spend a couple of months in the Microfit company. This is a French company in southern France. Fidi already mentioned the project that we worked together under the supervision of Professor Jean-Luc Mouget from Le Mans University. So this research stay was in the frame of this Ghana project related to marine diatom Haslea. So this company is business orientated, and they are delivering microalgal biomass and different bioalgal products. For example, here you see a patented formula called BrainFit, when they use fatty acids like omega-3 fatty acids from the diatom called Phaeodactylum tricornutum. This diatom is commonly present in phytoplankton all over the world, or in every marine environment, almost every marine environment. And they simply culture the biomass and squeeze the biomass to get these fatty acids from this biomass. And they patented a new nutraceutical called BrainFit. This BrainFit now is an ingredient for many products. You see here in American market, you see this food supplement containing this BrainFit formula. There are other products. For example, here you see vegan salmon fillet based 100% on diatom biomass. This is the species is called Odontella. Of course, you will find a lot of supplements, vegan omega-3 based on microalgal extract, but also a diatom extract. Why is like this? Well, the global market of microalgal products is expanding. Every year we have a huge money spent on microalgal investigation, but also on producing the microalgal products. Also among these are diatoms. Diatoms and microalgae, they are sourced not only for lipids, but also, for example, of vitamins and minerals. You see here, Haslea australia, so-called blue diatom, producing this blue pigment, which I will be telling you about. And it's very efficient in producing vitamin E, for example. And the industry, based on our basic research, based on the biochemical composition of this diatom, prepared a product, a cream for moisturizing the face. And you see 97 milliliter is sold for 30 American dollars. So it's a huge market and a huge money on the table. Diatoms are also a source of pigments. I'm not going to spend much time on this, but when it comes to diatoms, diatoms are famous for producing different kinds of beta-carotene, so the red ones or orange ones, with huge antioxidant properties. But also, they are even more famous for producing fucoxanthin, so the brown pigment, and diatoms, thanks to this fucoxanthin, diatoms are brown in color. And you see here a lot of diatoms producing huge amounts of this fucoxanthin, for example, Odontella aurita I mentioned, Phaudoctylum tricornutum, which you see here, but also recently there is one more promising blue pigment out of diatoms. It's called marinin. Probably you have heard about this because PhD is doing much of the research on this group of diatoms and on this pigment. But to those who didn't hear about this, so marinin is a water-soluble photosynthetic blue pigment discovered by accident, actually, because as you see here, in the late 17th century in France, they found that the oyster gills, they got bluish or greenish. Later on, the scientists looked under the microscope, and they found out that this blue color is because of the blue diatoms you see here. So we had and we have a lot of commercial aquacultures. You see here, this is Marin-Oléron region in Atlantic coast of France, where you see a lot of aquaculture ponds with these green oysters. But we used to have in Australia and Tasmania, England, Denmark, etc., because these green oysters are a conserve product. And they are much more expensive than traditional one. But when it comes to Haslea australia, it produces natural blooms. So we can monitor these blooms. We know where they grow. For example, I am doing in my research a monitoring of these blue blooms in the Adriatic Sea. So you see here my pictures from the Adriatic Sea from different years. Here is the coast of Montenegro, and you see a big pebble inside the water, and it's overgrown, or it's basically colored with the blue color. Here is a fragment of padina with blue patches on it, or a small rock or pebble, also taken from Adriatic Sea close to Croatia. Up to now, we know that only 5 species can produce this blue pigment, and this is Haslea specific pigment. So we don't see these, we didn't notice the presence of this pigment, marinin, this blue one, outside these 5 species and outside Haslea group. It has many possible applications. I presented you a face cream. So the food industry as well, but also possibly a pharmacy because of its antiviral, antibacterial, antimicrobial, anti-inflammatory activity. There are 2 types of marinin, one which is accumulated inside the cell, and at some point, these diatoms, they can release this maranine in the form of extracellular maranine, like you see here, a small bubbles, blue bubbles from under the microscope. And when they do that, the culture medium, you see, gets also greenish or bluish, and we call it blue water. This is this blue biodiversity when species was discovered and described from Java Sea. This is Haslea nusantara, but more famous one is Haslea australia. This is the first ever described blue diatom, and it was basically present in the northern hemisphere, Atlantic, Mediterranean. This is Haslea silbo from originally described from Canary Islands. This is Haslea provincialis from Mediterranean Sea, and one more Carthagensis from the Black Sea. So Haslea australia was basically found recently in the northern hemisphere. However, together with my colleagues from South Africa, we did a regular sampling along the South African coast. So this is the Indian Ocean, this is the East Coast, but also we did a West Coast, the Atlantic coast. And you see here that we were able to isolate a strain of blue-looking diatoms. In our eyes, it was very similar to Haslea australia. So we did quite a lot of research on this, and we also did phylogeny. And what we found out that this is actually indeed Haslea australia. So you see here over 99.5 identity with the different strains from Haslea australia. But I will show you this but later on. So fortunately, after a few days, we observed that this strain could also produce and accumulate maranine, so good for us. And after 7 days, we observed that the culture medium was also blue. So again, point for us, because we want to work on this maranine. So this is the COX-1 tree presenting different strains of Haslea. And this is Haslea australia, isolated for the first time in South Africa in the Indian Ocean. But also you see here on a different part of the tree, Haslea australia isolated from Australia. So what was fascinating to us when we did this molecular work, that our strain, the South African strain, can be discriminated. So it's quite distant from the Australian strain, but instead of this, it's associated on a tree and with the genetical identity with the strains from Atlantic French coast. So the area where the Haslea australia is present for centuries. So the question for us was, what were the driving mechanisms that we discovered closely related, genetically closely related, and morphologically as well, Haslea australia strain in a different part of the world, in South Africa, because we knew only that Haslea australia in Atlantic Ocean, for example, is present in the northern hemisphere. So this time we want to go a step further with our Haslea research. We submitted the project called Towards Sustainable Marine Biotechnology. So where we are going to explore the potential of Haslea strains for lipids and nanomaterial production. So we always start in our research with the basic research, with the basic knowledge, because we want to know how many different species and how many different population the Adriatic Sea hosts. So for the first, at the very first step, we are going to sample and isolate Haslea strains, and we are going to characterize them using a microscope like light microscope or scanning electron microscope, as well as genetical characterization to find out what is the exact population of these blue diatoms belonging to Haslea genus. Later on, we will go to a screening. So we will look for a candidates and conditions for a lipids production, and our aim is actually to explore the potential of this blue Haslea in extraction and production of these omega-3 fatty acids, APA and DHA. Then we will go, we don't want to waste anything from our biomass, so we will use the fresh gels to produce metal nanoparticles. So based on Haslea fresh gels and silica, we will be producing nanoparticles. Also, the plan is to test the properties of maranine as a stabilizing agent or reducing agent in the synthesis of these metal nanoparticles, for example, silver, gold, iron, and platinum. We'll see if marine has something to do with a reduction or stabilization of conditions when we will be synthesizing nanoparticles. And the most applicable part of the project requires testing these nanoparticles and testing biosilica in water remediations. So we are going to check if these nanoparticles and biosilica from Haslea is efficient in cleaning water from some chemicals. Okay, the next point in my presentation is related to diatoms and production of biomaterials. So another bridging point and integrative approach in diatomological science is related to ultrastructure of diatoms. With the scanning electron microscope, we can see a real beauty of diatom fresh gel. You see these ornamentations, you see these pores, you see this intricate, very complicated structure of diatom fresh gel. This is the surface. But some of the species, they have much more complex ultrastructure than we thought. So without basic research, we didn't know what candidates we can use, for example, for applied research. What you see here on the bottom images, this is the fresh gel, the cell wall of diatoms with a very intricate, very complicated infrastructure. So from the surface, from an outside view, you see this structure, but when you go inside, when you go external on the external part, you see totally different arrangement of the pore. The same as here. So this is a green light for us to test the ability of this fresh gel in, for example, water remediation. So for the first time, I thought that I may do something else than taxonomy when we prepared this publication. So the application of focused ion beam technique in taxonomy, oriented research on ultrastructure of diatoms. In this research, we did a scanning electron microscope, and later on, with the focused ion beam, we could perform a nanocuts of a diatom fresh gel to see how complex the diatom fresh gel can be in the structure. So we didn't know about some channels inside. You see this. We didn't know that there are some channels hidden in this fresh gel. So for me was a point, breaking point, if I may say that, to do something except the taxonomy in morphology. So I started to do my applied research as well. Another another article was about mathematical modeling of stiffness of the diatom fractal. So how hard the diatom fractal can be, what is the geometry of the fractal, and how we can use these models, these results from modeling in applied research. So with this, we are going to diatom biosilica. So we started to culture diatoms for a biomass and for metabolites and biosilica. So we have different systems of PBRs, photobioreactors. You see here very primitive one, 40 liters each, where we are able to produce a monospecific biosilica, 100% pure. It means this is only a hydrated silica, a monospecific, so one species. You see here on the screen, this is Melosira numuloides, so the capsules like fractals. So what we can do with this and what we did with this. For example, together with our colleagues from Białystok in Poznań, we start to develop a new biomaterials. In this case, I'm presenting you a biopolymers where we used a silica fractals of Halamphora salinicola. You see this crushed fragments of silica, and my colleagues added to this fractal PLA. This is a polylactide. This is a biodegradable biopolymer produced, for example, by a maize, by a corn. And we had we prepared a new biopolymer based on a diatom fractal. So what are the advantages of this kind of approach? First of all, it's a biotailoring. I can call it biotailoring. Biotailoring is not possible without a basic research. So we have to know what are the shapes of diatoms, what species is this, what is the size of the pore, what is the biology, etc., of this species. So the biotailoring is simply designing biomaterials according to the need. And with this, and with the basic knowledge about ecophysiology of these diatoms, we can manipulate the culture conditions and to produce the biomass according to the need. So if my colleague says, please prepare for me this and that size of diatoms, I am able to do this. So this is an example, a simple explanation of biotailoring. We can, by this way, we can produce environmental friendly and very cost-effective biomaterials, new biomaterials, which are sometimes an alternative to a traditional petroleum products. And sometimes they can even have more or better features. For example, this biopolymer had a better feature when it comes to thermoplasticity. So under a certain temperature, this biopolymer stayed a plastic. Of course, the huge advantage of this kind of bio-based biotailoring is the availability of diatom materials. So we can go to the sea, we can isolate diatoms, we can culture the diatoms, and then we can screen for a good candidate for a good shape of a fractal, good producer for lipids, good producer for pigments, and so on and so on. One more example about diatom biosilica is related to the production of nanoparticles. So diatoms are, and their fractal is kind of a scaffolding for a nanoparticle. So this is a reduction surface for some metal nanoparticles. So for example, we do in our research gold, silver, iron, zinc, titanium, germanium nanoparticles to using diatoms to produce nanocomposites with some certain features. For example, one of them, they can be good because they have present some antibacterial properties. This is basically related to silver. or photocatalytic or conductive materials, etc., etc. Why algae? Why we use algae and why we use diatoms specifically for the nanoparticle production? So, for example, when it comes to diatoms, this is a factory for nanosilica. I presented you the pictures with very intricate fractal, complicated structure of diatom fractal. Also, the diatoms and the fractal provides highly stable conditions for the reduction of metal particles. So there are three traditional ways of producing nanoparticles. This is the chemical, physical, and biological synthesis. We use the chemical synthesis, so we use the ions, the solution of various metal ions, and we add this ion solution to our biomass. And as I told you, diatoms act as a reduction surface for these nanoparticles. There are multiple uses of this kind of nanoparticles. Probably you have heard some of these places where we can apply these nanoparticles. Few years ago, there was a nice paper presenting application of diatoms and nanoparticles in so-called targeted drug delivery in a cancer treatment. So what you see here, this is a diatom fractal where the scientists put a lot of nanoparticles with a drug, and they implemented this to a human blood, and these nanoparticles and these diatoms you see here, they attached to the cancer cells. So this is a very precise way of delivering a treatment. What we do in our research with the biosilica, I already mentioned one phase of our new proposed project. This is a wastewater treatment. Because of this fascinating fractal, because of these pores, because of these areoles and this ornamented silica fractal, we see that diatom biosilica has a very high absorption and adsorption capabilities, and that's why we use this as a frame or a scaffolding for a nanoparticle production. It has, as I already mentioned and then shown you, a very and highly porous structure. So a lot of nanoparticles can be attached to this silica. So we are producing, or we produced actually, quite a lot of these nanoparticles. Here I'm presenting you nano silver metal particles. So you see here, we used a diatom called Gedaniella flavovirens, which was isolated from the Baltic Sea. Why is it important for us? I will show you in a minute. So this time we produced a silver-shaped hybrid nanoparticles based on Gedaniella flavovirens biosilica. In this case, on the other species, Gedaniella mutabilis, we produced the same type of metal nanoparticles, but with a different shape. You see here, these nanohybrids now are more spherical, more oval, not the flower-shaped. Why? Because we can manipulate with the conditions in the lab. So we can, for example, manipulate the pH of the reaction to form different shapes and different sizes of these metals. Another example, as this is iron oxide nanoparticles. We used, in this case, Phaeodicutylum tricornutum. I already mentioned several times during my presentation this species Phaudactylum tricornutum. This is this planktonic species, very famous for producing lipids, and also fucoxanthin. In this case, we used this very fragile frustule of Phaudactylum tricornutum to synthesize iron oxide nanoparticles. And these were spindle-like shape you see here, and they were containing a quite decent amount of iron. We then went step further, and we used this iron oxide nanoparticles in the degradation of nitrophenol, so the chemical compound nitrophenol. And you see here, within the time, the rate of nitrophenol degradation was very, very high. So almost 94% of nitrophenol degradation we achieved in the situation after 3 hours of reaction with these diatoms plus iron oxide nanoparticles. Here again, one more example from a gold nanoparticles. Again, we were able to synthesize different shapes of gold nanoparticles. Here you see triangular, they were spherical, or even some of them were hexagonal. Synthesized by Nanofrustulum schilloi, but also we used fucoxanthin, so the diatom pigment, brown pigment, to see if the fucoxanthin has something to do with the reduction. So by adding a fucoxanthin standard and extracted from this diatom species Nanofrustulum schilloi, we achieved a different sizes and slightly different shapes of these gold nanoparticles. So why not marinin? Let's check the ability of marinin, this blue pigment produced by Haslea, if the marinin has a reducing capabilities or stabilizing capabilities in a nanoparticles production. I told you that we also, our colleague Alexandra, performed her PhD on application of these nanoparticles. Here she tested an absorption rate of nanoparticles, different strains of Nanofrustulum. You see here different colors representing different Nanofrustulum diatoms, and the absorption rate of methylene blue. So different strains had a different absorption rate, but at the end, you see that this degradation, this absorption rate was very, very high. So the water, the culture medium was successfully removed almost in 100% from methylene blue. Again, I mentioned you about the pH and the manipulation with pH. Also here, we can manipulate with some of the conditions in the experiment to see what are the changes. And we see that at some points, in some situations, the removal rate can be slightly different, but in general, it's very, very efficient way and a green way in reducing removing the methylene blue from the solution. Alexandra actually did a lot of different tests. She tried a Congo red, crystal violet, and malachite green removal on different organic dyes. And she exposed nanofrustules of Halanphora salinicola, and she looked at the removal rate of these certain dyes within the time. And again, the same results, or more or less the same results. And the conclusion, the general conclusion, is that diatoms and diatom nanosilica especially doped with metal nanoparticles, is very efficient in filtrating, in removing some chemical compounds from the aqueous, from the liquid solutions. Why Baltic strains? We try to do the Baltic strains because of, if I may say that, problems with a Nagoya Protocol. I don't know if you have the same issue with the Nagoya Protocol in Indonesia when you do your research, but here in Europe and in Poland, we have quite big issue with the Nagoya Protocol. So we are not able to use any genetic resource, biological resource, natural resource from any other country without an agreement. And this is not the agreement for taking samples. This is agreement of conducting research based on mutually agreed terms. So in other words saying, what the country of the origin of this resource, like diatom strain, will gain from our research. Is it money or is it something not monetary? So every time we use something biological, genetical, or natural resource from any other country, we have to deal with the Nagoya Protocol and with these 3 different documents, which are very hard to actually get. That is why we are using more and more often, or actually nowadays 100%, we use the natural resources, so the diatoms from Poland, because Poland is not regulating yet the access to the genetic resources. So we can go to the Baltic Sea, we can collect the samples, and we can do a screening, do the basic research, morphology, biology, and then do the screening for some good candidates for lipids production, pigments, silica, etc., etc. In the few coming days, there will be a new paper published by some group of scientists from Poland, where we are trying to deal with the problem of unlocking the biotechnological potential of the Baltic Sea microorganisms, and also, of course, diatoms. So every time now I am checking the web page dedicated to Nagoya Protocol and access and benefit sharing. If you do some international research, I recommend you to do so, because, for example, Indonesia is regulating, quite strictly regulating, the access to the natural and genetic resources of your country. And Nagoya, so Indonesia is a part of the Nagoya Protocol from 2014. You have to first cooperate with some ministry. It's a Ministry of Environment in your case. Then do a research you want to do. Okay, thank you for your attention. I hope it was clear enough. I tried to cover a lot of various aspects, and if you have some questions, I'm very happy to stay here and answer your questions. Thank you very much.
Wow, thank you very much indeed, Przemek, for your inspiring presentation. It's really interesting, and me personally, I have a lot of questions, but well, I will let the audience ask first. So I am pretty sure that a lot of curiosity from our audience now, and we'll now begin the Q&A session. Yeah. And for the first segment, I would kindly invite the first 3 questions. So the attendees may use the feature of raise hand in Zoom, or type the questions in the Zoom chat box. Okay. So when I mention the name, please turn on the video and state your name and institution, and then deliver your questions. So is there any questions so far?
I see, I think one.
Yeah.
There's one question. How is the
Yeah.
treatment and equipment required for preserving and cultivating diatoms that you used for laboratory studies? So,
Thank you.
very basic, actually, equipment you need. You need a light inverted microscope. you need a good skilled person who will be will be isolating diatoms under this microscope. Well, the knowledge how to make a culture condition, sorry, not culture condition, but culture medium. And the diatoms are very opportunistic, so they can grow easily everywhere. So you don't need any sophisticated equipment. You can put them on the light on the window and they will grow. So just light microscope, I think that's the most essential one. When it comes to preserving, I don't know if I understand it correctly. So by preserving, you mean keeping the strains for a long time, I think. So we don't preserve diatoms like cryopreservation. We try to re-inoculate the diatoms every 2-3 weeks, 1 month to keep them alive. So we don't preserve them. If we see that something is interesting, then of course we grow them in a higher biomass. If something is not interesting and we have done all these taxonomy, systematics, we did a DNA extraction and so on, then sometimes and very often diatoms are simply dying because we cannot sustain huge amount of strains in our lab. We are only a few people working on live diatoms.
Okay, thank you very much for your answer, Przemek. So for the next question, someone here. Mbak Mariana, please, you may deliver your question, Mbak Maria.
Hello, good morning, Dr. Shimei. Let me introduce myself. My name is Mariana. I'm in phycology group led by VD, Mas VD. So I have, thank you for very intriguing presentation, and I was triggered with one slide that you show previously about the so there's a graphic about the comparison of lipid production and TAG, if I'm not mistaken, in a different environment. I mean, not environment, in a different
conditions and in the stress conditions. Yeah.
Condition. So you also mentioned earlier about the. I mean, it's triggered me because so I believe this all in a laboratory condition, right? I mean, I'm intrigued. I'm intrigued whether if let's say if I have if I want to not if I want to if I let's say if I have one one species of diatom which live in a normal environment, I mean in a field, and one is in an extreme environment. It's so from the graphic, it might be differentiate their ability to, their ability to produce lipid, right? So
yes.
yeah, so I mean, do you have any experience whether you compare two different sites, for example, with similar strain of diatom, for example?
So you can manipulate with the ability of diatoms in accumulation of the lipids. And these manipulations is this environmental stress. We call it environmental stress, but you simply apply these stress conditions in the lab. So you have your strain, you have a monoculture, and you do some series of experiments. And from my perspective, diatoms, they accumulate more lipids in a very bad conditions, for example, of light. So you can shade the light, or you can lower the amount of light you provide to them, and you can leave them for a while, for a few days, and you see if they react to these conditions. Because what we know is that this is a storage compound. So they produce diatoms. The diatoms, they produce lipids when there are bad conditions, and the lipids production is ongoing faster when these conditions are really bad to them. So first of all, what we do in our research is we are trying to find out what are the best conditions for these strains to grow, not to produce the lipids, but to grow. And then we can try to find out these conditions which are very bad for them. So, for example, it can be nutrients depletion in culture medium, or it can be a light. When it comes to the Baltic Sea, the salinity is around 7 per mile. It's very, very low. So we can also apply a salinity stress and see how they react and what kind of lipids they can produce. So, yeah. So first, the optimal conditions, and then see if we can somehow enhance them for a bigger production, for bigger accumulation of lipids, using different conditions not optimal for the growth.
Okay, thank you, Przemek.
Okay.
How about it, Mbak Mariana? Is it clear enough for
Yeah,
you?
yeah. Thank you.
Okay, thank you. Now we have another participant here. Bapak Hengki, please deliver your question.
Thank you. My name is Hengki. Actually, our culture and menstruality, which is diatoms or kids in similarity. In our experience, we have able to maintain the species on the strain of several microalgae, but by using manipulating the octopore formation in diatoms. I want to ask about how the diatoms will affect the size of diatom will affect the number or the amount of certain compound that you presented in this graph. Is there any correlation with size of the diatom? And is there any information that in your research about is there any condition of water quality that can cause several microalgae, especially diatoms, that they cannot grow in certain season? I mean, in my experience in culturing microalgae, especially Phytoceros, in parties, we face several times when the condition of the culture is not too good to reduce the carrot, but in certain times they grow without any difficulty. I mean, what kind of compound, maybe a trace element of in the water that can affect the carrot in growth?
Excuse me, sir. Excuse me, sir. I think your voice is not really clear for us.
Maybe you can ask the question on chat or email me, because
Yeah.
I barely hear you. So I would like to answer you, but I didn't hear you clearly. If you can write on chat or mail me, then that would be perfect.
Yeah, same with me too, because it's not really clear, Pak Hengki. Is it possible to write it down in the chat box, please, if you don't mind? Yeah. Yeah, okay, thank you. And we will try to find another question here from the chat box. There is
What's the good media for benthic diatom? I see the question from
yes,
Hengi.
from Henki, yes,
So,
please.
so what we use is, in our lab is F2 culture medium.
Okay.
It's why we use this, because it's very straightforward. The recipe is very straightforward. We don't need a lot of compounds. And these compounds are also not expensive, and it's very important to us because we use a lot of this culture medium.
Okay.
Of course, the best idea, the ideal situation is if we can use a natural seawater. We just need to filter them and maybe sterilize them, and this is the best idea. But I mentioned you that the Baltic Sea is 7 per mile, so it's very low salinity. Plus, it's very dirty. I shown you on the first slide these blooms. So we try to avoid using the Baltic Sea for the diatom culture. So we use F2 culture medium.
Okay. So F2 culture so far is the best medium for the benthic
Yeah,
diatoms.
it works for us. It works for us.
Yeah. Okay. Thank you for explanation. I hope it answer a question from Bapak Henki. Okay. And for the next question, we have from Befiona Helen. Hi, Mr. Dabek. I would like to ask, perhaps it was explained earlier that diatom lipids can be used as a raw material for vitamins that are already on the market. However, my current question is that I'm researching for my thesis about the characteristic of microalgae that have the potential to be used as biodiesel. And their content is more or less the same as what has been explained, namely that the presence of tag makes it one of the best materials for producing biodiesel with high quality lipids, and the presence of saturated fatty acids, or SFA, and monosaturated fatty acids, or MUFA, also makes it one of the ideal components in terms of chemical properties for biodiesels. I would like to ask whether biodiesel can be produced effectively without SFA and MUFA, relying solely on tag, and whether it is possible to obtain SFA and MUFA content from household waste such as coffee bean husk or cocoa bean husk. Okay, this is really interesting.
Very
Yeah,
interesting and
please.
very complex. So answering to this
Yes.
question, that's true. Diatom metabolites are present on the market. I've shown some of these products. There are a lot of these, not only for diatoms, but from diatoms as well, as vitamins, lipids, etc. The presence of TAC makes, of course, diatoms or makes any other algae very promising source for biodiesel. So I mentioned Botryococcus brownie, which is considered and used on a higher scale for producing biodiesel derived from algae. We had a problem. We had a project with our national oil company where we were asked to test the ability and the production rate of tags and lipids for a biodiesel purpose. And what I wanted to say and express, unfortunately, or maybe fortunately, diatoms are not very good candidates for biodiesel purpose. They accumulate very few amounts, very few percentage of tags, even under the stress conditions. When you compare, for example, diatoms and green algae like chlorella, because in our project we also tested chlorella. So chlorella, the green algae, was much efficient because of the higher growth rate at first, so the higher biomass, plus more tags accumulated in the cell. So I don't believe diatoms will be ever used in the biodiesel production. But I presented you different ways of using lipids from diatoms, like, for example, cosmetics. Unfortunately, as far as I know, we are not able to produce only tags in any microalgal group. So this is always a complex composition of different groups of fatty acids. I know that there were attempts of using well, how to say, genetic manipulations to like move or to shift the pathways of lipid production in the green algae and cyanobacteria, but I don't know the results. Unfortunately, I will not be good at answering to this part of the question. And I have no experience about household wastes, unfortunately. So I don't know if we are able to obtain fatty acids or not. I'm sure there are some remnants in the coffee beans or cocoa beans, but unfortunately, I don't have this knowledge. Sorry.
Okay, Przemek, thank you for your explanation. So I hope you satisfied with the answer, with Fiona. And we can go for the next question from Rios Tantika. Okay, from the Rios. Okay, I will read the question. Thank you for your insightful presentation. If environmental stress occurs naturally, for instance, under extreme conditions such as high temperature, salinity, or acidity, could it have any positive effect on diatom growth?
Okay, very interesting question. Is there any positive effect on diatom growth? Then I don't know, but there is a positive effect on accumulation of the certain metabolites. For example, we isolated once one strain of Haslea from a hot springs in Turkey, oleh. So in these hot springs, we saw that this Halamphora was able to accumulate much more lipids than any other strain of Halamphora. And we used this Halamphora strain for the lipids production. So definitely there is a kind of adaptation in the way that in some extreme environments, in the natural extreme environments, like for example hot ponds or hot streams or whatever, diatoms, they have the ability, like a secure system in the way they producing some metabolites, for example lipids. I wouldn't say it has a positive effect on the growth, but definitely we do screening. We should actually do screening by definition and bioprospecting, looking on extreme environments. For example, sea sponges living down at the bottom sea, right? They have some special adaptation. They must produce some special enzymes, natural products that we can use then, for example, for the cancer treatment. So by definition, bioprospecting is that we should look for the extreme environments and these adaptations to these extreme environments.
Okay, thank you very much, Przemek, for the explanation. So actually, me, myself, and Rios Tantika is collaborating now to study the diversity of diatoms in a geothermal region in hot springs.
Excellent.
So I'm really looking forward for the results of Haslea that recently isolated from Turkey. Yes. Okay, so for the next question, for the other segment of Q&A, we have Muhammad Untung Kurnia Agung, my colleague from University of Pajajaran. Okay, his question is, one common problem with the utilization of secondary metabolites from diatoms or microalgae is the instability of their production, especially when cultivated far from their natural environment. So how do you think this issue can be addressed? Thank you, Dr.
Very good question. So this, I think, in my eyes, this is the biggest obstacle that the microalgae are not ruling the world yet. This is this instability of, unfortunately, of culturing. I mentioned you that I had this research stay over almost over a year in Microfit, in the company related to production of biomass. They had a huge problem in sustaining a product to their customers because some microalgae, and we see also in our research that they grow sometimes faster, sometimes slower, depends on, I don't know, a time of the year. So unfortunately, this instability in biomass production is really a problem in upscaling the production, because we basically know everything about these commercially used strains of microalgae, and the same for diatoms. So I don't know. That's the question. Why they are so unstable in the growth, in the production, because the cultural conditions are the same. This when it comes to diatoms, it happens that simply from time to time diatoms are falling from the exponential growth. It means the strain is I don't know how to say, too old for some further cultivation and production of biomass. So we must re-inoculate them, wait for their best growth, etc., etc. So this is a biological system, unfortunately, and I don't know how to answer you to satisfy you with my answer, but definitely that's one of the biggest obstacle in biomass production.
Okay, thank you, Przemek, for answering the question. Yes, we still have a lot of homework for that.
Sure,
Yeah.
definitely.
So we go for the next question from Mas Danang Ambar Prabowo. He's actually a member of the Ecology Research Group in our research center. So thanks for your insightful presentation. My question is, if diatoms are known to produce more lipids under stressful temperature conditions, do you think they will similarly increase lipid production in response to future climate scenarios, such as higher seawater temperatures? If so, would this be considered beneficial?
Yes. Thank you for the nice inspiration for a new project proposal. But honestly, yeah, that's the very good question again. So what we do when we apply the temperature stress, so for example, if the optimal condition of a certain strain is, I don't know, 15 degrees, then we reduce the temperature, I know, every 5 Celsius degrees. And of course, diatoms can adapt to this situation over a time. So I think when it comes to the climate change, diatoms can adapt to this. And there will be, in the future world, in the future scenario, in the future global warming, there will be a new, they will be still the same. I mean, they will be simply adapting to these conditions, because these changes, although they are dramatically fast, diatoms are also fast adapting, even in the culture. So yeah, but we see, for example, in the Baltic Sea, that the composition of diatoms is changing because of the changes in the Baltic Sea related to the global climate change. So maybe that's the question, that's the case.
Okay, thank you very much for your answer. And we have another question from Henki again. Is the cell size of diatom could also affect lipid? Because when we culture the diatom, cell reduction were inevitable. Yeah, it's interesting. Reduce cell size along with cell division. Yeah.
Yes, of course. My colleague Eva Górecka, I shown you on the first slide, she is she did a research related to a certain family of diatoms, and how, and she was trying to answer how the reduction of size during, for example, a vegetative type of reproduction influence the accumulation of lipids. And there is a positive correlation. So, other words saying, if the diatom cells, they are shrinking in size, they are reducing their size, also the capability of lipid accumulation is lower. So what we want to do in the cultural conditions, we want to keep them in the exponential growth phase. So they are it means they are reproducing very fast. Of course, ideal situation if they will start to reproduce sexually, so to bring back the original size. But yeah, there is a positive correlation related to the cell size and the cell division, and the, as I told you, capability of accumulation, because simply the cell size is also decreasing. So theoretically, the place for lipid accumulation and storage is also lowering because of the cell division.
Okay, thank you again for your answer. And we have, okay, last question here in the chat box from Rahman Arif. Okay. Hi, Mr. Dabek. I was a bit late to this seminar, and my apologies if you have explained this in the presentation. But I was curious on why do you choose Halamphora to produce tag or fatty acid in your research, not other diatom species? Were they the best one in terms of metabolites production or biomass, or in terms of growth rate?
Sure. Okay, so why we have chosen Halamphora? Because we did a screening. So we had several strains in our culture, and in controlled conditions, in the optimal conditions, we saw that this certain strain accumulates a lot of lipids. So we decided, okay, let's check how this will react. This strain will react in the stress conditions. If the accumulation of lipids will be increased or decreased or the same, whatever. So at the first, we do this kind of a screening. So we look on diatoms and diatom strains, and we see how they are acting in the normal standard conditions. And then if we see if they accumulate a lot of lipids in the standard conditions, it means they also, or it can be like this, that they will also accumulate more and more of these lipids in under the stress conditions. But it's not going it's not happening every time like this. Sometimes they are surprising us, and even if the diatom is producing low lipids in the normal conditions, they can rapidly increase this lipid production in the bad conditions. So this is a matter of repeating experiments, actually. Also, answering to the second part of the question, there is no key which species, which genera will produce more lipids, which one will be producing more pigments, etc. This is, I would say, a blind process. So you have something in the culture and you test it.
Okay. This is really interesting. Maybe I have a follow-up question based from Rahman question. So did you find different trends between same species that found in certain region with another region? For instance, I personally myself found that the antibacterial activity of Haslea australia and Haslea nusantara very much different to one specific bacteria. Yeah, and is it the same case for the lipid?
So we didn't, yeah. I understand what you mean, and this is very interesting to see. But we didn't test yet the ability of, for example, lipids production. Let's stick to this. with the same species from the different regions. First of all, we didn't have the same strains, the same species and different strains from the different regions. Because of this Nagoya Protocol, we really want to stick to the Polish natural resources to omit these problems, this bureaucracy with these permissions. And of course, Baltic Sea has a very limited amount of biodiversity in terms of diatoms. So we didn't check what is the production. Are there any differences in production of, for example, lipids or any other compounds in the same species from the different regions? We didn't do that yet.
Okay, thank you very much for your answer. So I think we have no more question in the chat box. So maybe I should return to Mbak Gloria. Hello, Mbak Gloria. Should I be the one who close the session or?
If someone will have any other question later on, then please mail me or forward this question to FIDI. I will be happy to assist you or help you in answering.
Okay. And yeah, maybe while we are waiting for Gloria's response, I'm really interested in what you presented before about the blooming of Haslea in like macroalgae, thallus, something like that. Did you observe in the Baltic Sea as well?
No,
Or
we only have Haslea, non-blue Haslea in the Baltic Sea.
Okay, non-blue Haslea.
So
Okay.
yeah, we have 2 or 3 different species of Haslea, but non-blue. They are not producing pigments.
And this non-blue Haslea is already described, or
yes,
because
it is described. Yes, it is described. Yes.
All right. Okay, thank you. Okay, so Gloria said that I can close this session. So, Dr. Shemek, thank you so much for your excellent and inspiring talk. And we do have a lot of what do you call knowledge and very inspiring like publication from you, your research achievement, and we wish you a very good luck for your project in the near future.
Thank you very much.
Yeah, and on behalf of the entire Research Center for Biosystematic and Evolution in BRIN, I would like to extend our deepest gratitude to you. Yeah,
thank you
for the incredibly insightful and inspiring presentation. So we do hope that we can collaborate in the near future. So maybe you can come to Indonesia to do like visiting
I will be very glad.
Research, yeah, or a postdoc program from our management Talenta from BRIN. I think, yeah, it'll be interesting for us to collaborate in the future.
Sure,
And
thank you.
so
thank you for considering me as a speaker. Thank you. Thank you very much.
Thank you. Yeah. Thank you as well to all the attendees, our researchers, scientists, and students as well for your engaging questions. It is very interactive session, Q&A session, and this concludes our session. So have a wonderful day, everyone, and have a nice weekend. Yeah, and see you in the future, maybe. Yeah,
yes.
I hope.
Thank you, guys.
I hope.
See you in Indonesia.
Thank you, Shemek.
Thank you.
Yeah.
Terima kasih.
Bye.
Thank you. And Shemek, we will
Sampai
send.
Pak Fidi dan Bapak Ibu sekalian.
Terima kasih, Bapak Ibu. Ya.
Terima kasih.
Shemek, maybe we will send you a certification for your talk today. Okay, in a few weeks, I hope.
Okay.
Yeah, and also small souvenir from our research center.
No,
Yeah.
it's really not necessary. I was very
No.
happy
We are happy. We're happy to do it. Yeah. I'm more than happy
Okay.
to do it, actually.
Thank you.
So I see you later, and please send my regards to Romang and other faculty members in your lab.
Okay,
Yeah.
sure.
Thank
All the
you.
best.
Yeah.
Bye.
Bye-bye.
Bye.
Okay, thank you everyone. Izin untuk live.